Today we will discuss about isolation of actinomyces trees producing antimicrobial compound from different soil sample collected from medicinal plant area. So introduction in antibiotic actinomyces trees consist of an extensive and diverse group of gram-positive bacteria. These look like just as fungus, and then these are the literature follow which I have followed for my research, like Deepka et al. 2009 isolated 100 actinomyces trees, and Manjula C et al. In 2009 isolate 6 of the actinomyces trees, and then Ojgar C et al. In 2008 and Igarci Y et al. And then aim and scope of the research: the need of new and useful compound to provide assistance and relief in all aspects of the human condition is ever growing. So screening of microorganisms for the production of novel antibiotics has been intensely pursued for many years of scientists. Antibiotics have been used in many fields including agriculture, veterinary and pharmaceutical industries. The research will be very useful for the future study for the screening of actinomyces from the soil sample. Then plan of birth: first collection of sample, then isolation of actinomyces, and then classification of actinomyces, and then isolation of antibiotics, actinomyces producing antimicrobial compound, and then extraction, and then at last purification of the compound was performed. And then these are the material method. Like first collection of sample, the different soil sample were collected from different parts of India, and then isolation, these soil sample were diluted, and then these were spread on the different media, and then certain test microorganisms were selected for the antimicrobial activity, and then characterization of isolated actinomyces from sample were performed. At last, production of antimicrobial compound by using algae broth and starch casein was performed on the temperature of 37 degrees centigrade. So the extraction, when I performed different experiment, then I found an A1 and AR2 shows some activity against the microbes, and these are the result shown in the slide, like RIS1 and RIS2, which shows Rosa indica soil, and then SIS1, SIS2 and SIS4, which shows Sarca indica soil, and then aloe vera soil. And these are the plant which I selected from where the soil were collected nearby these plants area. And then gram staining of the microbes were performed, and then these are the results for, as we can observe in slide, like ISP2, ISP4, ISP5 and 7 tests were performed, and then these are the biochemical tests, and then at last IR, NMR and mass spectroscopy of the compound were performed. And as we observed in our slide, these are the soil sample, and then these are the actinomyces which were showing in the slides, like yellow, orange and white color colony. And here photograph number 11 shows antimicrobial zone of invasion, and then these are the photograph for TLC, which shows blue spot on the TLC, and then these are the paper chromatography slide, and these are the compound which were extracted, and these are the value for UV, IR, NMR and NMR of the compound. So here we observed there are two compounds were produced by the actinomyces, A1 and A2, and these are looking like 2N-butanamide, 3-methyl, 4-methoxy, 5L-aronisoil, propanophenone and sterosporin like compound. So these study will be very useful for the future, because when I performed some antimicrobial activity of these compound, the zone of invasion was found more than streptomycin against some of the microbes as observed. So So at last, thanking you.